So can you hear me? Tejas?
Yes, sir.
ओके, तो लास्ट क्लास में हमने डिस्कस किया था ट्रांसक्रिप्शन यूनिट एंड द जीन। अगर हम बात करें ट्रांसक्रिप्शन, द प्रोसेस विच द formation of the synthesis of the RNA from the pre-existing DNA known as the transcription. And at the time of the transcription, only one strand act as the strand of the DNA act as the template DNA. यहाँ पर रीजन है why the both strand does not copy the transcription. Simple answer is if both strand act as the template, there would be RNA molecule different sequences and the different sequences of the proteins. Okay, consequences हो जाएगा ये. Hence, one segment of the DNA would be coding and for the two different protein. And second reason is the two RNA molecules produced simultaneously would be complementary to each other. Hence, would forms a double-stranded DNA, which is the stable DNA as compared to the single-stranded DNA. That's why only one strand of the DNA act as the template strand at the time of the transcription. Because of if the two RNA molecules RNA produced simultaneously, then form according to the complementary principle, and then forms a double-stranded DNA. Double-stranded is a type of the stable DNA. And when the stable DNA, so translation does not take place. So now हमने डिस्कस किया था फिर ट्रांसक्रिप्शन यूनिट, the three units in the promoter, structural gene and the terminator. Promoter region where the transcription start, structural gene where the actual transcription take place, and the terminator where the transcription terminate or the stop. Then and during the transcription, DNA dependent in RNA polymerase involved, and the catalyze the polymerization only in the one direction, which is the fifth prime to the third prime. And the strand that has the polarity, which polarity 3 to 5 act as the template strand, and which has the polarity, इसको क्या बोलेंगे, template strand बोलेंगे. The other strand which has the polarity 5 to 3. इसमें जो RNA जो फॉर्म होगा किस पे होगा, 3 to 5 वाले बहुत RNA का जो सीक्वेंस होगा 5 to 3 होगा, तो द एंटीपैरेलल होगा अकॉर्डिंग टू द कॉम्प्लीमेंट्री प्रिंसिपल. एंड इन द केस ऑफ द RNA, instead of the thymine, the nitrogen base will be type of the uracil. Strangely, this strand which does not code for anything, which strand does not code for anything, known as the coding strand. इसकी जो polarity वो क्या होगी, 5 to 3. All the references point, ये point बहुत important है, very important. All the references point while defining a transcription unit made with the coding strand, such as like the coding strand is a type of the reference point that we can explain or the define anything in the transcription unit according to the coding strand, such as the location of the promoter. So promoter located on the fifth strand of the coding strand. Therefore, the location of the terminator will be third prime of the coding strand. And if the positioning of the promoter and terminator exchange, the terminator to promoter यहाँ पे आ गया, then the strand will be reversed. ये वाला क्या हो जाएगा, template strand, this one is, and this one should be will be coding strand. And here is the promoter and the transmitter flank that the structural gene in a transcription unit, the both side of the promoter and transmitter, both side of the structural genes exist करते हैं. And the promoter is said to be located toward the fifth end of the structural gene. So mainly हम डिस्कस करेंगे यहाँ पर ट्रांसक्रिप्शन यूनिट एंड द जीन. Gene is the defined as the function unit of the inheritance. First point ये बताना चाहिए gene क्या. What is the gene? Gene is the defined as the functional unit of the inheritance. But in the define a gene in the terms of the DNA sequence. That the DNA sequence which coding for the tRNA or the RNA molecules also define a gene. And the cistron. Cistron means the segment of the DNA coding for a polypeptide known as the cistron. And the eukaryotic type of the cell में which type of the cystonic present? Monocystonic. And the prokaryotic में polycystonic. In eukaryote, the monocystonic genes have the interrupted by the coding sequence. Just the अगर हम बात करें, if we talk about the coding sequence of the express sequence type known as the exon. And exon are said to be those sequences that appear in the mature RNA. Always remember that exon regions of the sequences type of the coding, or we can also say that the appear in the mature or the processed RNA. The exon are interrupted by the intro. The intro or intermediate sequence do not appear in the mature processed RNA and do not code anything. Only exon code something. And then the influence of the intermediate sequence, like inheritance of character is affected by the promoter and the regulatory sequence of a structural gene. So today we will talk about the types of the RNA and their processes of the transcription. If we talk about in the bacteria, three major types of the RNA present: mRNA, messenger RNA, messenger RNA, tRNA, transfer RNA, and the rRNA, ribosomal RNA, which is the important in the metabolic role at the time of the transcription. So all three RNAs are needed to synthesize a protein in a cell. And the function of the RNA will be like the RNA responsible for the translation. And the translation means the synthesis of the proteins. That the mRNA provide the template, and tRNA brings amino acid that reads the genetic code. Very, very, very important one. What is the role of the tRNA? The role of the tRNA is brings the amino acid and then read the genetic code. What is the genetic code? We will discuss after some time. And the RNA plays structure in the catalytic role during the translation. At the time of the translation, metabolic role या catalytic role जो होगा वो किसका होगा? rRNA. RNA polymerase. Let's see first this point. DNA-dependent RNA polymerase that catalyze transcription of all types of the RNA in the bacteria. Like in the transcription, which type of the enzyme involved? DNA-dependent RNA polymerase. And catalyze the transcription, the formation of the RNA of the all types of the RNA from the one strand of the DNA. एक strand से क्या होगा? tRNA, mRNA, and the rRNA can be synthesized. And when the RNA polymerase, this RNA is the DNA-dependent RNA polymerase, binds to the promoter and the initiate transcription unit. The when RNA polymerase binds to the promoter when RNA polymerase binds to promoter and initiate the transcription. This process is known as the initiation. In it, use the nucleoside triphosphate as the substrate. And the polymerase in a template depend fashion following the rule of the complement. And complement पे हम पढ़ चुके हैं, such as the adenine only combine with the uracil and the cytosine only combine with the guanine. Between the two hydrogen bond and the three hydrogen bond respectively. And also facilitates opening of the helix in the continued elongation. Only a short stretch of the RNA remains bound to the enzymes that the RNA polymerase reaches the terminator region. The nascent RNA, the single molecule of the RNA, nascent means the single, like nascent oxygen, single atom of the oxygen, nascent hydrogen, such as the single atom of the hydrogen. Such as here, nascent RNA falls off and the RNA polymerase. And this results in the termination of the transcription. अब जो इम्पोर्टेंट है इसको हम डिस्कस करते हैं फिर देन interviewing point point is the how is the RNA polymerase able to catalyze all the three steps, such as the initiation, elongation, and the termination. Three basic steps in the transcription: initiation, elongation, and the termination. And when the RNA polymerase is only capable to catalyze the process of elongation, it has to share transitively with the initiation factor, which is known as the sigma factor. The firstly, we talk about the transcription. Firstly, RNA polymerase bind with the initiation factor, which is known as the sigma factor. Bind with the RNA polymerase and then bind to the promoter, and the initiation of the transcription will start. Such as the transcription, initiate, the terminate, and the transcription respectively. Association with these factors alter the specificity of the RNA polymerase to initiate or the terminate. When RNA polymerase binds with the initiation factor, such as the sigma factor, then the transcription start, and then the hydrogen bond will break. And जैसे RNA polymerase आगे move होगा, then hydrogen bond breaks, and then from the previous hydrogen bond which breaks during the transcription rejoin again, and such as the RNA length of the RNA will be increased. In the immature type of the RNA, both type of the sequences, such as the exons and the introns, will be exist. Like bacteria, since mRNA does not require any processing to become active. Which type of the processing we will talk about after some time. Like transcription and the translation take place in the same compartment. There is no separation of the cytosol and nucleus in the bacteria. And if we talk about the bacteria, talk about the transcription in the bacteria, transcription starts during the transcription. Translation will be start because of the no any type of the nuclear membrane present around the nucleus. That means the chromosome directly present to the cytoplasm. And the transcription DNA from the RNA and the translation protein synthesis from the RNA take place at the same compartment. There is no separation of the cytosol. That means the cytoplasm and nucleus in the bacteria. Many times the translation can begin with much before the mRNA is fully transcribed. But before the completion of the transcription, many times translation can start before the mRNA fully transcribed or completed transcription. And as in like the eukaryotic cell, two different type, two additional complexities gives me, such as there are at least 3 RNA polymerase. How many RNA polymerase? 3 RNA polymerase in the nucleus in addition to the RNA polymerase found in the organelles. There is a clear-cut division of labor. Specific curve होगा. And then RNA polymerase 1 transcribes, important point, transcribes rRNA, ribosomal RNA, 28S, TNS, and the 5'S. Whereas the RNA polymerase third is responsible for the transcription of the tRNA, 5'S RNA, and 5'S small region RNA, and the small nuclear RNA, small nuclear RNA. In the RNA polymerase second transcribe precursor of mRNA, and the heterogeneous nuclear RNA or the hRNA. This type of the heterogeneous nuclear RNA. And the second complex bit, that is the primary transcript contain both the exon and the introns are the non-functional here. When the mRNA will form at the time of the transcription from the single strand of the DNA, then some type of the processes are required for the survival of the RNA into the cytoplasm, because of the when RNA moves from the nucleus to the cytoplasm of the cell, then required protection from the lipase enzyme, which enzyme can destroy the RNA. For the protection from the ligase enzyme, RNA should ongoing from the some type of the processes such as the splicing, capping, and the tailing. Splicing, if we talk about the splicing, where the introns are removed and exons are joined in a defined orders. This process is known as the splicing. Heterozygous under RNA undergoes additional process called the capping and the tailing. If we talk about the heterozygous type of the RNA, at the time of the heterozygous RNA, capping and the tailing. In capping, unusual nucleotide methyl guanosine triphosphate (NGT) added on the fifth end of the heteronuclear RNA. And in tailing, adenine residue attached to the adenine group attached to the how many units? 200 to 3 units, 300 units are added at a 3 end in a template independent. If we talk about the manner, it is the fully processed RNA. The significance of such complexity is now beginning to the understood. The split gene arrangement represents probably an accident feature of the genome. The presence of introns is the reminiscence of the antiquity. And this one is the RNA. Genetic code. Now we will talk about the genetic code. During replication and transcription, nucleic acid was copied to form the another nucleic acid. Like if we talk about the transcription, replication, synthesis of the DNA from the pre-existing DNA, transcription is synthesis of the RNA from the pre-existing DNA. And these processes are easy to conceptualize on the basis of the complement. In the process of the translation required transfer of the genetic information from a polymer of the nucleotides. Genetic codon. During replication transcription, a nucleic acid was copied to form the another nucleic acid. Thus, these processes are easy to conceptualize on the basis of the complement. In the process of the translation requires transfer of the genetic material from a polymer of nucleotide to synthesize a polymer of amino acid. Neither does not any complementarity exist between nucleotides and the amino acids. Process of the translation requires transfer of genetic information, like polymer of the nucleotides. And neither does any complementary exist between the nucleotide and the amino acid. And if we determine the biochemical nature of the genetic material and the structure DNA was very exciting. The proposition and the differing of the genetic code were most challenging. In a very true sense, it required from this point, if determine the biochemical nature of the genetic material and the genetic structure of the DNA was very exciting. The proposition and the deciphering of the genetic code when most challenging. Okay, most challenging task है, and it required involvement of the scientists for several disciplines. Physicists who argue with the since there are only four species, and if they have to code for the 20 amino acid, the code should be constituted a combination of the base. We should that in the order of four for all 20 amino acid. The code strand made up of the three nucleotide. This is very blurred whole proposition because of permutation combination of the 4 power 3, 4 into power 4 into 4 into 4 would generate 64 codons, generating many more codons. So providing proof that the codon was a triplet and a more daunting task. The chemical method developed by the H. Bovin Corona by the instrument in the synthesis RNA molecule with defined combination of phases like homopolymer and the copolymer. The Marcel Niederberg cell-free substance for protein synthesis will finally have to control the pollution. And such enzyme is the Severococa enzyme, polynucleotide phosphate. Polynucleotide phosphate. And was dust in polymerization reacts. Polymerization. These are the genetic codon. Uracil cytosine adenine in the guanine. Uracil cytosine adenine in the guanine. इसको हम जो समझेंगे लाइक फर्स्ट वाला यू यू सेकंड वाला यू हो गया एंड थर्ड तो यू one u is here and the other one is the here. How many u? 3u. That means the if it call the UCU, U C in the U. You see you answer in the the straight features of genetic code are as follow. Remember these points. The codon is a triplet. 16 code for amino acid, 3 does not code.
Sir, voice is not clear.
आवाज नहीं आ रही?
वो क्लियर नहीं है.
एक मिनट. Right now, right now,
Yes, perfect.
okay. So now talking about the silent feature of the genetic code, and as the follows, such as the codon in the triplet form. Always remember that the genetic code will be in the triplet form, and in the 64 total codon, 61 codons code for the amino acid, and 3 codon does not code for amino acid. Well, only out of 64 codon, only 61 codons code for the amino acid. Only 3 codons do not code for anything, and then the known as the stop codon. And some amino acids are coded by more than one codon. Those type of the amino acid coded by the more than one codon. Hence, the codon is known as the degenerate. Like example की अगर हम बात करें, like leucine, same protein है, same amino acid है, पर इसकी जो codon है वो क्या different है. Like CUU also called the leucine, CUC also called the leucine, CUA also called the leucine, CUG also called known as the leucine. मीनिंग क्या बोलते हैं इन कोडोन को? विच amino acid coded by more than one codon known as the degenerate, and the codon is read in mRNA in a contagious fashion. They are the no punctuation, and the code is nearly universal. Code will be universal. For example, from bacteria to the human, capital U, 3U, which code for the phenylalanine. And some exception to this rule have been found in the mitochondria codons and in the some protozoans. इनमें the codon क्या है? Universal है. For example, bacteria, human में U जो triple U होंगे, code करेंगे इसको phenylalanine. And some exception भी यहाँ पर. Then AUG has a dual function. It codes for the methionine amino acid, and it also acts as the initiation vector. Initiation codon. So just stop codon की हम बात करेंगे तो what is stop codon? UAA, UAG, and UGA are also stop terminator codon. So right now, here AUG इसको कोड करेगा मेथिलीन को. Capital U 3 times code the phenylalanine. UUC phenylalanine. UUC so AUG and the triple U and the UUC and AUG code the methionine. And all sequences genetic codon code the phenylalanine. What हो गया? Degenerate codon. Degenerate codes. And mutation in the genetic codon. Let's see about the syllabus. in the genital codon. Is everything clear? Deep. Can you hear me?
Yes.
Okay. Now we will talk about the protein synthesis. Like to get a DNA, you get a chromosome structure in the gene and the regulation of the gene expression. So mainly we will talk about the protein synthesis. So now we will talk about the translation. What is the meaning of the translation? Translation refers to the process of the polymerization of amino acid to form a polypeptide chain. The process of the polymerization of the formation of the synthesis of the amino acid from the to form the polypeptide chain. In order to end sequence of the amino acids, are defined the sequence of the base in the mRNA, and the amino acids are joined by the bond which is known as the peptide bond. Between the amino acids, when the polypeptide chain form, then the between the amino acid bond will be formed is the will present the peptide bond. Formation of the peptide bond requires energy. What required? Energy required at the time of the formation of the peptide bonds. Therefore, in the first phase itself, amino acids are activated in the presence of ATP. In the first phase itself, amino acids are activated. This point is important. Charging of the TRN जिसमें हम बात कर रहे हैं. Amino acids are activated in the presence of ATP and linked to their cognate type of the tRNA. When the amino acid activated in the during the presence of the ATP and bind to their cognate. Cognate means the their type. Then after the amino acid bind to the ATP, then the bind to their own type of the tRNA. Own type, that means the cognate tRNA. And process commonly known as the charging of the tRNA or the amino acylation of the tRNA. To be more specific, if two such charged tRNA are both close enough, the formation of the peptide bond between them take place. This is a very energetic process. The presence of the catalyst would enhance the rate of the peptide bond formation. And catalyst from आप पहले से जानते हैं, like as you already know that the catalyst, those type of the molecules can enhance the rate of the chemical reaction. Like cellular factory responsible for the synthesis of the protein. And cellular factory is the ribosome. Cellular factory responsible for the synthesis protein is known as the ribosome. So the ribosome consists of the structural RNA. And you already know about the components or the subunits of the RNA or ribosome. The subunits of the ribosome will be two types. One large subunit and the one small subunit. And if we talk about the large subunit in the eukaryotic type of the cell, 60S, and the small subunit will be 40S. And always remember that the ribosome attached to the mRNA. And if we talk about the which subunit of the ribosome attached to the mRNA, always remember that the small subunit of the ribosome attached to the mRNA. Is it clear or not? They just deep. Can you both listen me?
Yes, sir.
So let's see here. Cellular factory responsible for the synthetic protein in the ribosome. The ribosome consists of the structural RNA and about 80 different proteins. And in its inactive state, it exists as two subunits, large subunit and the small subunit. When the small subunit encounters the mRNA, the attached to the mRNA, the process of the translation of the mRNA to proteins begins. And there are the two sites in the large subunit. Subsequent की अगर हम बात करें, the two sites in the large subunit for subsequent amino acids. In the two sites in the large subunit for subsequent amino acid to bind to the thus be close enough to each other and to formation of the peptide. Let's see here the tRNA. The mRNA, the base sequence of the mRNA is here, and the tRNA comes and the read the genetic code and the brings the amino acids again and again. And when the two amino acids are close to each other and then forms the peptide bonds. And the ribosome also act as the catalyst 23S RNA in the bacteria in the enzyme, such as the ribosome, to the formation of the peptide bond. In a transcription unit in mRNA is the sequence of mRNA. It is flanked by the start codon. The translation unit in the mRNA. Start codon, a very important point. Start codon is the AUG, which is called the methionine type of the amino acids. And the stop codon codes encodes for the polypeptide. And mRNA also has some additional sequence that are not translated. So the mRNA also have the some type of the sequences which does not translate into the anything, such as known as the untranslated region UTR. And the UTR present on the both end, at the both fifth end and the before the start codon and the third codon after the stop codon. They require for the efficient translation process. Like for initiation, the ribosome bind to the mRNA at the stop codon. Oh, stop nahi, start codon AUG, and that recognized only by the initiator tRNA. The ribosome, firstly, what will happen? Like at the time of the initiation of the translation, ribosome bind to the mRNA, and then cognate type of the tRNA recognized, is known as the initiator tRNA. And then ribosome proceed to the elongation phase of the protein synthesis. Elongation phase, that means the translation, formation of the polypeptide chain. And during this stage, complex composite of the amino acid linked to the tRNA and sequentially bind to the appropriate codon in mRNA by forming complementary base pair. According to the complementary base pair, like अभी हम डिस्कस किया जेनेटिक कोडोन. जेनेटिक कोडोन के 3 ट्रिपलेट फॉर्म में होता है. जो ट्रिपलेट फॉर्म है जेनेटिक कोडोन, उनके अकॉर्डिंग क्या होगा, टीआरएनए उन कोडोन को रीड करेगा एंड देन द ब्रिंग द एमिनो एसिड्स. The ribosome moves from codon to the codon along the mRNA. And the amino acids are added one by one, translated into the polypeptide sequence, dictated by the DNA and represented by the mRNA. That means the translation mainly dictated by the DNA. Because of the replication to the transcription to translation. It is a journey for the formation of the proteins. And at the end, the release factor binds to the stop codon, terminate translation, releasing the complexes polypeptide from the ribosome. ये आज समझ में आया नहीं आया? जल्दी बताओ सब।
Yes, sir.
ठीक है, आज के लिए इतना ही। कुछ इमरजेंसी में मुझे कुछ काम है थोड़ा सा। नेक्स्ट क्लास में डिस्कस करेंगे रेगुलेशन ऑफ द जीन एक्सप्रेशंस। ठीक है, सो यू कैन एंड राइट नाउ।